lapel.
Learning her principles of biochemistry. Chapter 1: The Foundations of Biochemistry. Evolutionary Foundations, Part 2. The first cell probably used inorganic fuels. Have you ever wondered how life began? Scientists have been exploring this question for centuries, and recent discoveries have shed light on the origins of life on Earth. Around 3.85 billion years ago, the Earth was a very different place. The atmosphere was nearly devoid of oxygen, and microorganisms were scarce. But somewhere in this early environment, the first simple organism arose. This first cell, or protocell, likely used inorganic fuels such as ferrous sulfide and ferrous carbonate, both abundant on the early Earth. These inorganic compounds provided the energy necessary for the cell's survival and replication. The reaction of inorganic compounds like ferrous sulfide and ferrous carbonate yielded enough energy to drive the synthesis of ATP or similar compounds, which are essential for cellular function. Some theories propose that these inorganic compounds may have originated from extraterrestrial sources, such as space dust containing simple amino acids and organic compounds. This process of using inorganic fuels to drive cellular functions and replication was a crucial step in the evolution of life on Earth. It laid the foundation for the development of more complex organisms and the diverse forms of life we see today. The study of the first cell and its use of inorganic fuels provides valuable insights into the early stages of biological evolution. By understanding these fundamental processes, scientists continue to unravel the mysteries of life's origins. Eukaryotic cells evolved from simpler precursors in several stages. Have you ever wondered how the complex eukaryotic cells that make up all plants, animals, and fungi came to be? Around 1.5 billion years ago, the fossil record begins to show evidence of larger and more complex organisms, probably the earliest eukaryotic cells. These cells marked a significant leap in complexity compared to their prokaryotic counterparts. Details of the evolutionary path from non-nucleated to nucleated cells cannot be deduced from the fossil record alone, but morphological and biochemical comparisons of modern organisms have suggested a sequence of events consistent with the fossil evidence. Three major changes must have occurred during this evolutionary process. First, as cells acquired more DNA, the mechanisms required to fold it compactly into discrete complexes with specific proteins and to divide it equally between daughter cells at cell division became more elaborate. Second, as cells became larger, a system of intracellular membranes developed, including a double membrane surrounding the DNA. This membrane segregated the nuclear process of RNA synthesis on a DNA template from the cytoplasmic process of protein synthesis on ribosomes. This was the evolution of the nucleus, a defining feature of eukaryotes. Third, early eukaryotic cells, which were incapable of photosynthesis or aerobic metabolism, enveloped aerobic bacteria or photosynthetic bacteria to form endosymbiotic associations that eventually became permanent. Some aerobic bacteria evolved into the mitochondria of modern eukaryotes, and some photosynthetic cyanobacteria became the plastids, such as the chloroplasts of green algae, the likely ancestors of modern plant cells. Molecular anatomy reveals evolutionary relationships. The key to understanding the molecular foundations of evolution lies within the DNA, the genetic blueprint of life. DNA is a long, thin, organic polymer composed of deoxyribonucleotides, the building blocks of genetic information. The linear sequence of deoxyribonucleotides in DNA encodes the instructions for producing proteins, each with a unique three-dimensional structure. This process is essential for the functionality of proteins, which play a crucial role in the diverse functions of living organisms. Comparing gene sequences across different organisms reveals remarkable similarities, indicating a shared ancestry and evolutionary relationships. These homologous sequences provide valuable insights into the fundamental unity of life. Living organisms are open systems, constantly exchanging matter and energy with their surroundings. The intricate processes of cellular metabolism involve a dynamic steady state, maintaining a delicate balance between energy consumption and production. Entropy, a measure of randomness and disorder, plays a critical role in biological systems. From the randomization of heat to the oxidation of glucose, entropy shapes the dynamic nature of living organisms. Functional genomics shows the allocations of genes to specific cellular processes. Functional genomics field focuses on understanding the functions of genes and their interactions within the context of a whole organism or cell. Through large-scale studies, scientists have been able to determine the allocation of genes to specific cellular processes. This means that we can approximate the proportion of the genome dedicated to a specific process, such as membrane transport or protein synthesis. Genes encode the proteins and RNA required for various cellular processes. These proteins play crucial roles in the structure and function of the cell, and their interactions are essential for maintaining the cell's integrity and carrying out its functions. Furthermore, the regulation of gene expression is vital for ensuring that the right genes are activated at the right time. This intricate process allows the cell to respond to its environment and maintain its internal balance. The insights gained from functional genomics have increasing importance in medicine. Large-scale studies have allowed the identification of genes in which mutations correlate with various medical conditions, offering potential targets for drug therapies and personalized medicine. In summary, functional genomics provides a comprehensive understanding of how genes are allocated to specific cellular processes, shedding light on the intricate mechanisms that drive the functions of living organisms and offering promising avenues for medical advancements. Genomic comparisons have increasing importance in medicine. Genomic comparisons involve analyzing the entire genomic sequence of different organisms to understand their genetic makeup and evolutionary relationships. Each organism's genome contains a wealth of information encoded in the sequence of its DNA. By comparing these sequences, scientists can identify genes associated with various medical conditions. Genes, which are specific segments of DNA, play a crucial role in the regulation of cellular processes. Large-scale studies have allowed the identification of many genes with mutations that correlate with medical conditions. For some genetic diseases, these genomic comparisons have paved the way for potential cures, replacing palliative treatments with targeted therapies. The study of genomics also provides insights into the evolutionary foundations of life. It's fascinating to think about the origins of the first cells and the role of RNA and proteins in the early stages of life. The RNA world hypothesis suggests that RNA may have been the first genetic material and catalyst, playing a crucial role in the evolution of early life forms. Moreover, the maintenance of genetic continuity over millions of years is a testament to the remarkable stability and precision of the genetic material, DNA. The genetic information encoded in DNA provides a historical record of the long journey from the earliest cells to modern organisms, shedding light on evolutionary relationships and the fundamental similarities shared by all living organisms. In conclusion, genomic comparisons have become a cornerstone of modern medicine, offering great opportunities for understanding the genetic basis of diseases and conditions, and paving the way for personalized treatments and interventions. Thanks for watching. Don't forget to like, share, and subscribe for more fascinating insights into the world of genomics in medicine.
PharmaPal.